Awesome.
Great.
Well, I'm sorry I can't be there with you all today. I wish I could. I always enjoy coming to Southern Missouri and hanging out with Patrick and Kelly and all my buddies down there. But unfortunately, with the rain and the fact of the USDA making like every grant proposal in my life due tomorrow, it just became impossible for me to get away from Kansas City. So anyways, we're going to talk today a bit about grafting for tomatoes mainly. And some of this stuff may be things you all have probably heard before.
And then we do have some new stuff to talk about as well too. But kind of my hope was just to really provide an update about, all the different types of research we've done at K-State related to grafting and how it could be, hopefully effective for you all at your farms. So as probably many of you know, I work at the Olathe Horticulture Center just outside of Kansas City, and this is a picture of some of our high tunnels. We do definitely have a focus on high tunnel production in my research program. There was a time when we did a lot of work where we would compare high tunnels versus open field production and look at quality and shelf life and things like that. And we still do some of that. But most of the work we do is really focused inside the high tunnel. So what are ways that we can develop new crops? We can increase yield. We can increase the economic profitability of these high tunnel systems. This is a little time lapse shot that I took just the other day. We covered our last high tunnel on our big hay grove about 2 weeks ago. And so let's see if I can get this thing to work. It goes by quickly, but it's kind of fun when it does. So that's us covering the tunnel. I don't know if any of y'all have played with drone technology, but you can make the coolest videos, all kinds of fun stuff. Thankfully, there wasn't any wind that day. That wasn't the case. We usually do 2 bays at a time, and the bay next to it, we got it halfway up there and the wind just started kicking like crazy. And so unfortunately, the week before we just did 1 bay, so we only had to do 1 for our last one. So probably as many of y'all know, the grafting of vegetables is not really an unusual thing when we get outside of the United States, and it's becoming more popular and sort of commonplace here in the US as well too, especially for high tunnel production specifically. But there's very large nurseries worldwide. These pictures are from Korea of grafted watermelons and tomatoes and eggplants at this nursery. This is a nursery I just visited. I guess it was before the lockdown, so I guess it was 2 years ago now. And I'm actually planning to spend part of my sabbatical this next coming spring working with these folks. This is Agris greenhouses in Greece, and they actually produce about 15 million grafted plants per year, and they export them to Romania. They use them in Greece and Crete and other parts of sort of those Baltic region. And they have a very large scale production system that they use for grafting plants. Now, about half of the plants that they graft there are watermelons, and the other half are tomatoes. And if you didn't know, watermelon grafting has really increased in popularity here in the US in the last really just 3 to 4 years. Excuse me. So there's all kinds of fun things you can do with grafted plants, and we won't really talk about any of that today, but they do offer, I've noticed you can buy online these grafted plants that have multiple scions, right? So this is one that I grafted a million years ago when I was in graduate school, and it had there was a German Johnson scion as well as a Cherokee purple scion, and then grafted onto a Beaufort rootstock. And in this case, it was an interspecific hybrid rootstock. To help increase that vigor and stuff. So, this isn't really something a commercial grower would ever do unless you wanted to grow these grafted plants and sell the plants themselves at the farmer's market. It is remarkable what some of the seed companies get for these double grafted plants. I mean, I think they're upwards of $20 per plant. So as I mentioned before, we really focus on grafting with tomatoes, and we have published some work with peppers as well. We did a couple years of pepper trials in our tunnels, but we haven't really worked too much with cucurbits. But I will say that I think there's a lot of value in looking into grafting cucurbits, and that could be cucumbers. Oftentimes for high tunnel production or greenhouse production, people will graft cucumbers onto squash rootstocks because they're more cold tolerant. Or like I mentioned before, watermelons are really increasing in popularity in terms of use of grafted plants, and not just for, small scale growers like us here in Kansas and Missouri, but out in California, they're planting like, hundreds of acres of grafted watermelons because they're trying to deal with fusarium wilt out there. And that's one of the things that you can manage with rootstocks. Okay, so as I mentioned before, when we graft tomatoes, we typically graft them onto what's called an interspecific hybrid rootstock. So that's a cross between a wild and a domestic species, essentially. And what that rootstock does is it really brings across some disease resistance as well as just yield, sort of vigor, I guess I should say, that in order to increase yield for those plants. And so, there's rootstocks, there's lots of research out there of different rootstocks that can be utilized to manage certain diseases, or on the abiotic side, things like salt, soil salinity, and some of those kinds of things. But the challenge with research is we have to be able to identify which rootstocks are useful for which issues. So that keeps us busy here at the Olathe Horticulture Center, and hopefully will probably keep us busy for a little while. So what are the benefits of grafting? Well, as I mentioned before, most of the rootstocks that we're grafting with are going to be interspecific hybrids. So they'll oftentimes have resistance to certain soil-borne pathogens that isn't even available in commercial, non-grafted lines. So a good example of that's for a disease that occurs around here is one called southern blight. And that's called, it's caused by a pathogen called Sclerotium ralphii. And Sclerotium ralphii is a fun fungus I did a bunch of work in my PhD on, but it definitely occurs around here. And unfortunately, there's just no horticulturally acceptable varieties that have any good disease resistance to it. But we have found that many of these rootstocks actually have some moderate disease or intermediate disease to that resistance to that pathogen. So that can be a really useful thing because if, there's lots of resistance out there available for Fusarium wilt race 1, for example, but not for some of these others. And then, of course, the root system is a physical anchor. It's also what's going to be bringing up the water, nutrients. So there's lots of evidence in the research that shows that some of these really vigorous rootstocks can just bring up more water and nutrients and really supply the upper portion of the plant what it needs for increasing that yield. So we've done a fair amount of work looking at economic feasibility and also economics of producing grafted plants. And, I didn't really want to go through piles and piles of data as we talk about this, but there's two main themes that have shown up when we start talking about profitability and economic feasibility for using grafted plants. One of them is the system that you grow them in, right? Whether it's going to be a high tunnel system or a field system. And at the end of the day, it doesn't really matter what system you grow them in. What matters is that it's an intensive system where you're looking for high productivity. And of course, every grower says they want high yields, but the reality is a mature green tomato picked in Florida 3 times or even 2 times over the course of the harvest season is going to yield a lot less per plant than a high tunnel heirloom variety that gets picked all the way from the month of June until October, right? Just because of the intensive management of that. And so, one of the things that we find is in order to make grafting more economically feasible, you have to be able to increase the yield to offset the cost of the grafted plants and hopefully make some money on that investment, right? And so if you're not yielding very well because of the system, and open field is a good example of that, then you're not going to be able to increase, that revenue in order to really pay for the plants. The other thing that I think is probably more important in this conversation is the sale price. If you're selling your fruit for 20 cents a pound or 10 cents a pound in 25-pound boxes, then the reality is, you're just never going to be able to make up the dollars and cents that it takes in order to pay for that plant. Now, we've published data that shows you can buy a plant from a nursery for about $1.60 and that will give that nursery a 50% markup, which is pretty typical for most of the sort of small scale propagators out there. Similarly, you can grow your own plant for about 79 cents to $1, somewhere in there. So if you need to make that dollar, plus you want to make some money back on that dollar that you've invested in it, you know, you need to be able to increase the poundage, but most importantly, you got to be able to sell that fruit for a good price in order to pay for it. So, you know, as a result, and it's really more correlation than causation, we see a lot of high tunnel growers drifting over into grafting technology earlier than others, mainly because they're selling their fruit for a high enough price that they can afford it. So that's sort of what this slide shows, is just sort of all the different systems that people have looked at grafting in. And when I was in graduate school, we did a lot of work looking at grafting as an alternative for methyl bromide in these very large scale systems. And again, some of that economic data, what it shows is just, you know, for those really large scale growers, they just don't sell that fruit for high enough, and they don't get enough pounds per plant to really make it worth their while. And we've found that with the processing industry. We just did a big project through our SCRI grant with a bunch of folks over at UC Davis that work with the tomato processing industry, and they were actually already buying grafted plants. But once we started doing the research with them, they figured out that, you know, it just really doesn't pay for itself. There's a lot of folks here in the area that are interested in using grafted plants. This survey data is getting a little bit old, and my guess is some of these numbers have changed. But one of the things that I wanted to point out in this slide was, you know, when we first got into this work of grafting and growing grafted plants, we sort of had this vision that all these, you know, small scale vegetable growers around Kansas and Missouri would start grafting their own plants and in order to, you know, just like they grow non-grafted tomato plants, they'll just grow grafted ones and then plant those in their tunnels in their open field. And what we've learned since then is that, you know, the reality is it is kind of difficult to grow grafted plants, right? And oftentimes you have to have a nicer greenhouse, more different climate zones within that greenhouse so you can manage that plant growth. And so it can be really challenging for a lot of our vegetable growers to actually grow their own plants. And there's a lot of folks that are interested in buying them. And that's sort of what this data reflects. Now, I oftentimes get questions by folks, you know, if we're using these disease-resistant rootstocks, does that mean that we can stop using rotations in a high tunnel? Because obviously we know how important crop rotation is for managing diseases. But it becomes very challenging in a high tunnel from an economic perspective, because, you know, the intention is we need to make as much money as we can in order to pay for that structure. Now, I certainly understand that viewpoint, but I get concerned when I start talking to growers that, you know, are just constantly growing tomatoes after tomatoes after tomatoes in their high tunnels and have this notion that if we start using rootstocks, then they can continue that, you know, bad behavior. The reality is, at that point, you're just, you know, not rotating rootstocks instead of not rotating non-grafted plants. And there's actually been a lot of research done in Italy and other parts of Europe where they've been using grafted plants for longer than us, and they're oftentimes not rotating there either. And what tends to happen is they suddenly start having issues with pathogens and diseases that attack the rootstocks, right? Maybe they don't attack a non-grafted plant, but there's this funky Phytophthora they found for cucurbit rootstocks that only goes after the rootstocks. It's not a problem with non-grafted plants. So, you know, I really encourage you to think about using grafting as a way to supplement your rotation and not as a way to replace it. And I think there's a few ways to think about this. I've given several talks on sort of the economics of crop rotation. We won't get into the details of that today, but just to let you know, some of those videos are out there on YouTube. There's one on eOrganic posted there if you're interested in talking a little bit more about the economics of rotation. But one of the things that we like to do is thinking about that revenue in the long term. So if each one of those crops is going to generate a certain amount of revenue per square foot, you can put together a 6-year crop rotation that includes, you know, tomatoes once or twice, and you can still have a good revenue per square foot over that 6-year period. But you have to be able to plan that out, and quite frankly, understand that, in this case, year one, we were getting $4.75 a square foot. Well, when we utilize the rotation, now we're getting $4 or $4.09. So you will reduce your revenue a little bit, but in the long run, you're going to increase the sustainability of that system. Okay, so let's get back to grafting itself. I wanted to talk a little bit about the propagation of the grafted plants. I think most of y'all know we use what's called the tube or splice grafting method, and there's lots of information out on the web about how you do that. We've produced a number of videos and sort of publications and stuff. We're actually getting around to updating a publication that should come out later this year as well from K-State. But essentially, these plants are grafted when they're pretty small. You can see the picture here. We do like to graft them below the cotyledon leaves. It's important to do that so you don't end up with suckers that come off the rootstocks. And then this little silicone clip just goes over the seedling stem, and then this would be our scion tissue, or the upper portion, the fruit-producing variety gets inserted there. We want to have these nice parallel lines that show that clip's going to match up really well. And you can see, in that case, we did a pretty good job making those cuts, so there's no gap in between them. We've done research with and also just played around with lots of different healing chamber types. If you want to do a small number of plants, these little domes work really well. Now you want to be careful about those, especially in a greenhouse. You can see we use this foam to just keep the direct sunlight off of those chambers. And I've been using every year, I think I use more and more foam in our healing chamber room. This stuff is really useful for if you have a day when it just gets real sunny real fast, and all of a sudden those plants start to wilt, you can just black them out real quickly with a little bit of that reflective foam board. Now, one of the challenges that we've run into over the years is this issue with adventitious roots that can come off the scion. And that can be a problem for a lot of different reasons. One, if you get enough of roots, like you see here in the middle of this picture, it will actually sometimes push the scion out of the clip, and a plant that seemed like it was healed and doing fine will actually become an unsuccessful graft. So that can be really problematic. But then the other issue is that if you are in a situation where you're trying to utilize a resistant rootstock to manage a disease, you've now just created this green bridge down to that infested soil from your susceptible scion. And so whatever benefit you get of disease resistance, you're not going to be able to exploit that because that pathogen is going to have a direct route to that susceptible scion tissue. And these are actually some pictures from that operation in Greece, Agris. They also, you may have seen, they actually have healing chambers indoors where they use LEDs, and this is pretty common for those large scale commercial nurseries. But they also have an adventitious root problem too. So I was, sad for them, but happy for me that I wasn't the only person that ran into that problem when I got a chance to visit with them. And that's actually one of the reasons I'm going back for my sabbatical in the spring, is to help them work on this issue. So we did some research a couple years ago looking at how we graft the plants affects the development of those adventitious roots. You can see up here in this top right-hand corner, if you get just a little bitty adventitious nublet like that, once the plant has healed and you move it into a lower humidity environment and out of that healing chamber, oftentimes those roots will just stop growing, or sometimes they'll even just dry up and sort of disappear. But if they get the opportunity to grow while they're in that healing chamber, that's when things can become really problematic. Now, I won't get into all the physiology of it today, but one of the things is we had this suspicion that if we were able to remove the leaves off of the plants, the scion, before we graft them, then that could affect the adventitious roots. And that's mainly because auxin, a plant hormone related to adventitious root formation, is created in the meristem of the plants, and it flows down the stem. And so we had this idea, maybe if we cut those leaves down a bit, we can reduce the amount of auxin that's going to the graft union and therefore eliminate some of that adventitious root issue. So in this case, you can see in this picture at the bottom, we had one group where we removed 90% of the leaf tissue. So we really hacked them off pretty good. One that was 50%. And then this is sort of your full leaf, or really 0% leaf removal. And here's the data from that experiment. And we did this a few different times. This was in the humidifier chamber. So this was under the highest humidity conditions. And when we didn't remove any leaves, we found that actually almost 20% of the plants were unacceptable in regards to their adventitious root formation. So in reality, you sort of have an 80% success rate in that situation. As opposed to the 90% leaf removal, we had 0 unacceptable plants and just a very few that had some adventitious roots, but they could be managed with a little bit of trimming and with the right environmental management. So we saw this very stark contrast. The more leaf removal, more leaf tissue we remove from that scion, the lower the incidence and the severity is of those adventitious roots. We also wanted to make sure that that process wouldn't hurt the growth of the plants once they get moved into the high tunnels. So we did basically some greenhouse studies to look at that. And what we found essentially was that there was, in the case of the leaf removal system, there wasn't any penalizing effects in terms of the growth of the plant, and more importantly, the first initiation and formation of the first flowers. All right, so I'm going to show you a video here. This is sort of what we've been up to a little bit more recently in the last few years. Welcome to the Kansas State University Olathe Horticulture Center. In the last 3 years, this little greenhouse outside of Kansas City has contributed nearly 250,000 pounds of tomatoes to the local food system. Surprised? Here's how. The use of grafted tomatoes is expanding across the United States. Research has shown that certain rootstocks can increase crop yield, and this technology is particularly useful for high tunnel growers. Many tomato farmers in the Midwest want to purchase grafted plants. Unfortunately, there are no local plant suppliers currently grafting. K-State started custom grafting for local tomato farmers in 2016. They use a technique known as splice grafting that was first developed in Asia. The rootstock and scion must be the same size when it's time to graft. Therefore, seedling uniformity and plant health are extremely important. The stems of the rootstock and scion are cut at a 60 to 75 degree angle. And then silicone clips are used to hold the stems together while the graft union heals. An experienced worker can graft approximately 150 to 200 plants per hour. Leaf removal increases grafting success and transplant quality as well. Once grafted, the plants are moved into healing chambers for 9 to 10 days. After the union has healed, the two plants are ready to grow as one. Since 2016, K-State has grafted over 20,000 plants for farmers in the region. They've also been collecting data from grafted plants at local farms. K-State researchers can estimate that those plants have yielded an additional 223,200 pounds of locally grown tomatoes to consumers. Now that's a lot of maters. All right, well, it's a little bit cheesy, but it is a lot of maters. And actually, I updated those numbers just before signing on here this afternoon. So since 2016, we've actually grown now 44,000 plants, because that video was made a couple of years ago, which is about 500,000 pounds of tomatoes. So, you know, the reason I bring that up is not really to brag about it, although we're obviously proud to have that kind of an impact on the community and for our growers. Obviously, you know, that many pounds of tomatoes is going to be good for growers too, right? But I think what's more important is I really just want to show that there's a real opportunity here. in the Kansas City region, I'm sure in southern Missouri as well. Anywhere where people are popping up high tunnels, there's an opportunity for people to grow these grafted plants. And I'll probably talk about it a little bit later, but we're submitting a proposal tomorrow through this large collaborative group that I work with, led out of NC State and Ohio State. And we're hoping to actually turn this program into sort of an apprenticeship. So if you know any propagators, and this will be an online apprenticeship, so somebody living in Springfield or Mount Vernon would be able to participate in this through Zoom and Facebook. And, you know, we're hoping to provide a really rich experience for some of these greenhouse operators in the region so that they can think about starting to grow some of these grafted plants, because we're pretty much maxed out on what we can grow here at the Olathe Horticulture Center, but there's a lot of opportunity out there for other propagators to do that. And if there was a propagator here in the area, I would happily pass along, you know, any information we have to help them get established as well. All right, so let's talk a little bit about rootstocks and high tunnels. So we've done a lot of work over the last many years, I should say, both in our high tunnels and other ones around the state, and some open field trials, really just trying to evaluate rootstocks. And I'm sure many of y'all have seen some of this data before, but we try and do as much on-farm research as we can, although we're doing less of that here in the last couple of years. This is a Gehringer's orchard in Edgerton, Kansas, and we're not doing a trial with Gehringer's this year because, quite frankly, they just have no use for the non-grafted plants. They've converted all of their high tunnels over to using grafted plants. And so, you know, this trend has been a little bit, it's a great thing for us as in research and extension, but it means I need to go find someone else to do trials with. But we've been able to generate some really nice data through Gehringer's and our farm and others to identify some rootstocks that do well here in the Midwest. Now, one of the things that I sort of work under the general assumption is that most growers in our area, at least in Kansas, don't really have a lot of issues with soil-borne plant pathogens. Now, I say that, and of course, somebody's going to call me tomorrow with a big issue, but most, you know, other than verticillium wilt race 2, we do have some growers that we're starting to see nematodes more and more, both I think because of lack of rotation in the tunnels, and also they're just becoming more prevalent in sandy soils, it seems like. But other than that, you know, those are our two main pathogens we're dealing with, and they're pretty low incidence. So one of the things we're always trying to do is identify rootstocks that increase yield when there's low disease pressure, because those are going to be the ones that are vigorous enough to increase our yield, increase our profitability, and put some money back into your pocket as a grower. So this list on the left in yellow, these are the ones that have really done well for us over the years. Maxifort, Multifort, DRO 141TX, Arnold, and Colossus. The Colossus they pulled off the market a couple years ago, and Arnold can be found, but it's difficult to be found. But there's also some new ones that have been coming out as well that we've been testing. To be honest with you, even the new ones don't seem to, you know, hold up to Maxifort and Multifort. Those are ones that have been around for a while, but in our trials, they're the ones that most consistently increase yield. And here you can see some of those newer rootstocks we've worked with. So Cardia is one, and you can see actually pretty good yield boost there. Fortamino, Supernatural, and some others. Supernatural is a rootstock that was actually exclusively for the Mighty Mater program. If you ever saw tomatoes being sold under that brand name, Mighty Mater, for a long time they wouldn't tell us what rootstock it was they were using, and then they figured out they couldn't make any money selling grafted plants for $20 a piece, so they started selling the rootstock seed and called it Supernatural. Although it wasn't available this year, I'm sure you all know just as well as I do, getting seeds for anything this year have been really difficult. So after a while of doing all these rootstock trials, year after year after year, we started putting some of this data together. And some of you may have met David Lowen a couple years ago. I believe he came and gave a talk at this same growers school, maybe back in 2018 or 17. And one of the things that David did was he actually took all this data from, like, I don't remember the exact number, but 30 or 40 different trials, and we put them into one big database and started looking at, you know, trying to identify what rootstocks consistently increase that yield and consistently show that added vigor. So here you can see some of that data, and I won't get into all the details of what we did here, but essentially the idea is this, and I think this is a useful lesson for growers, although there's a lot of math there. On this x-axis, this is called the production potential, and basically what we did was we took, out of these 30 or so different trials, we said, what is the highest yielding treatment, whether it's a grafted plant, a non-grafted plant, it doesn't matter. Tell us what the highest treatment is in each replication, and that's the production potential of that little square footage of space, essentially. And then we said, we took the actual yield data from all of the plants that were normalized to per plant data, and then we normalized them with the non-graft. So what that means is, if in a particular trial, Maxifor produced 20% higher yield or 30% higher yield than the non-grafts, then its relative productivity ratio was 1.2 or 1.3. So for example, this dot right here that's on 1.5, that means the Maxiforts produced at 50% higher yield than the non-grafted plants. Similarly, there's two dots down here at 1, right? And in those two replications, actually, it meant there was no yield difference between a non-grafted and a Maxifort. Then we said to ourselves, okay, we can characterize the environment because we know that the highest yielding plants are producing this much. So that means, the data over here were in trials that were overall doing better than the trials that were over here. And what we found was there was actually correlation with these two rootstocks, Arnold and Maxifort, that showed that as you increase that production potential, you actually increase the relative benefit of grafting. So what that means is those grafted plants are more efficient at utilizing those resources than the non-grafted ones are, because as the overall productivity of the whole plot went up, they actually went up at a higher rate, okay? So in those cases, we're labeling those as vigorous rootstocks because they're able to utilize those resources, whereas maybe some of these other ones don't. So on the bottom, we have RST-04106. That's a rootstock that's bred for bacterial wilt control and is used primarily in the Southeast US. And what we found was the exact opposite trend, right? We didn't really see a lot of increase in yield with that rootstock. And as you increase the yield of the overall trial, we saw even less benefit of using that rootstock. So we feel like this was kind of a cool way to identify some of these vigorous versus non-vigorous plants in terms of rootstock potential. And David was able to publish this, but it's I think it's kind of neat stuff because it gives us the ability to predict whether or not those plants are going to be useful in a certain system. But the take-home lesson for it, and after this long diatribe about math, is that if you want to capture the benefits of using those interspecific hybrid rootstocks, you need to give them the resources in order for them to succeed. So, there was a time when I was younger and sillier that I thought, oh, we don't need to fertilize grafted plants. They've got big roots and they'll find all the nitrogen that's in the soil and they'll survive in sort of more stressful environments. And that may or may not be true, but if you really want to capture the benefit of those rootstocks, and what you want to do is you want to fertilize them more because they can use it. So as I mentioned before, there's a lot of good information out there about the sort of the disease management side of some of these rootstocks, and I encourage you all to look at vegetablegrafting.org. We've got huge tables on that website with all the different rootstocks that show different resistance to different pathogens. But what we've really been focused on here at K-State was just trying to figure out this plant vigor thing. And so we feel like we're starting to get a feel for which rootstocks really provide that yield increase, even when there's not consistent disease pressure. Okay, so let's switch gears a little bit and talk about scions now. So this is a trial that we did in 2017 and 2018 looking at the combination of different scions with one rootstock. So usually in our trials, we'll have one scion, and we'll have a whole bunch of rootstocks that we're looking at. But in this case, we did it the opposite way. We had one rootstock, we had a whole bunch of different scions. And so these are all red slicers. I'm sure many of you recognize these variety types. BHN 589 is one of the more popular varieties for high tunnel production. We've been really impressed with Primo Red over the last few years, and also Red Deuce. And a lot of our growers that participate in our custom grafting services get Primo Red and Red Deuce as well. So this is sort of the raw data, just looking at the per plant yield. And you can see for some of them, and Primo Red's a great example, we didn't really see much increase in yield when we grafted those plants compared to other ones like BHN 589, where there's this huge jump in yield when it gets grafted. So we took that a step further and we said to ourselves, okay, well, let's look at the actual change in marketable yield in terms of pounds per plant. And you basically are just seeing that these purple bars are the distance between the two bars before. So you're going to see what happens there. But what's more fun is to look at the gray bars, because what that represents is the percent of the non-grafted productivity. So how much did we increase yield? So in the case of BHN 589, it was almost a 50% increase in yield, whereas Primo Red, it was just under 10% increase in yield. So we start to see these big differences when we start to normalize that data. So what we've done now is we've taken these scions, and again, these are really from our variety trials, and have put them into these general categories that we call highly compatible, moderately compatible, and compatible. And what that means is basically, it really has nothing to do with the grafting union success. It has to do with the success of the plant itself. So anything where we saw over 40% increases in yield, we saw that scion as being highly compatible with Maxifort. And these are a little bit arbitrary in numbers. Obviously, the difference between Richmond and Red Deuce is not very much, but sometimes things just sort of work out that way. But we're also, the good thing about data like this is we're also finding varieties that aren't compatible. So if you really want to grow Primo Red or Scarlet Red, then you may or may not be interested in using grafted plants. Okay, so we're going to touch a little bit on fruit quality. I'm fortunate to be located here near the K-State Olathe campus, where Dr. Pliaconi has a post-harvest physiology lab, and we've done a lot of work in the last few years looking at nutritional quality of tomatoes from grafted plants. So this was some of the early work that we did, just sort of looking at organoleptic quality, and that's basically quality related to flavor. So the Brix, or the sweetness level, and the acidity. And then oftentimes we'll look at that titratable acidity to Brix ratio as well, because the combination of the acidity and the sweetness is really where you get the flavor of the tomatoes. So when we utilize some of these vigorous rootstocks like Maxifort, it's very typical to see yield increases, but we wanted to know how's that going to affect the flavor. So we did not see any differences in Brix, but there were differences in acidity, and actually the Maxifort does have a little bit more acidity to it. Now, that doesn't mean it tastes worse. Oftentimes, people like acid flavored tomatoes, so it's a little bit of what your preference is, but there is a little bit of difference in that titratable acidity when we start looking at some of these high-yielding rootstocks. The other thing that was really interesting to us was looking at the antioxidant capacity. So this is the nutritional quality of those plants, and antioxidants are responsible for basically cruising around your body and getting rid of free radicals and things that can cause damage to cells in the human body. And what was interesting is there's this sort of assumption, I think, amongst a lot of horticulturalists, that as we find technologies to increase yield, unfortunately, that will result in produce or just crops in general that have lower nutritional quality. And so we wanted to find out if that was the case for antioxidant capacity. We have a high-yielding rootstock in Maxifort, and then we have these lower-yielding rootstocks in RST 04106 and RT 1028. And what turns out to have happened is that the Maxifort, the high-yielding rootstock, actually had similar nutritional quality, similar antioxidant capacity to the non-grafted plants. So that means that using these high-yielding rootstocks did not penalize the nutritional quality of the fruit. And that was evident in both 2014 and 2015. So we feel pretty good about those results because they're consistent. But then what was really interesting is, as it turned out, the low-yielding rootstocks, and again, these are rootstocks that basically produce as much fruit as a non-grafted plant, they actually had higher antioxidant capacity. So they had more nutritional quality than the non-grafted plants or the ones grafted onto Maxifort. So we got kind of excited about that because that's sort of a unique phenomenon that we don't see very often, finding ways to increase yield. Typically, we assume that that's going to reduce the quality. So we got really busy the last 3 years and started working with this scion called Tasty Lee. And I'm sure many of y'all know what Tasty Lee is. It's a high lycopene tomato variety. We had included in our scion trial and found that it really liked to be grafted in terms of increasing yield. We saw some of the highest yield increases with Tasty Lee that we had amongst all 10 of those varieties, in the neighborhood of 35 to 40%. So we grafted Tasty Lee onto some of our high yielding rootstocks and our low yielding rootstocks in order to look at how that affected not only the yield, but also the quality as well. And so Tricia Jenkins is a PhD student that's been working on this. Here you can see the yield data. I know there's a lot of numbers and letters there. I'll give you the short version, which is it wasn't as exciting as we were hoping for. In general, these plants yielded very poorly. It's pretty common for us to get 20 pounds of plant, if not more, in our high tunnel trials. And these unfortunately did not do that. So this trial we did in a movable high tunnel. It was new to us in 2018, and I'm not 100% sure if it was because of the soil where the tunnel was or because of something about the plastic, but for some reason, those plants just did not yield very well consistently for 3 years. And so we just didn't see a lot of spread in the yield data, which isn't really surprising when we think about some of the other trends we've seen. But Tricia's she's actually done all the lab work for the nutritional quality, and she's writing it up and doing the stats analysis now. And she's been looking at not only antioxidant capacity, but also lycopene and some of the other carotenoids that are really important for nutritional quality. So the story isn't done yet, but because I'm stubborn, we're actually replanting this study in a different high tunnel this year, because I just want to make sure that I'm not crazy and that Tasty Lee can yield better than it did in that movable tunnel. Okay, so we're going to wrap up here in just a few minutes, but I did just want to highlight some of the more kind of fun projects. This isn't as much grower related, but I think it's just kind of interesting. So we worked with a group in plant pathology who actually, Karen Garrett used to be at Kansas State University and then moved to University of Florida, but she took all of our DNA with her over there so they could continue that work. And essentially what they were doing was looking at the microbial community on the surface and inside the roots of the plants. And these were it takes a pretty long time to do all the lab side of this stuff. So you can see these were from early trials, but they were conducted in our certified organic high tunnels. And what we found was actually that the communities of microbes that live on these rootstocks is actually very different than the ones that we see on non-grafted plants. And there could be a lot of reasons we could speculate for that. But, it's kind of interesting that, this notion that we can start to utilize rootstocks to cultivate or manage these microbial communities, and vice versa. We can also think about breeding rootstocks for these diverse communities that might exist. And one of the things we did see was actually that especially Maxifort, our sort of vegetative rootstock, vigorous rootstock, it actually supported a higher diversity of microbes on the surface of the roots and inside the roots as well. So, that's sort of an interesting finding for a biology nerd like me, but I think especially, for organic growers in the world of grafting, we have a lot more to learn about all the interactions that happen between microbes in the soil and plant performance. So just to sort of wrap things up, grafting is definitely here to stay. I mean, we're producing 10 to 12,000 grafted plants per year for growers just sort of within a short drive of Olathe, Kansas here. And as people continue to grow in high tunnels and become more experienced in high tunnels and start to run into problems in high tunnels with soil-borne diseases, you're going to see that technology stay. Now, I'm not sure that it's here to stay for open field production, especially in the world of tomatoes. But for high tunnel production, it really is. And the reason for that is, most high tunnel growers are getting a pretty high price for their fruit, right? Now, we have found that certain rootstocks can really increase yield. And I showed you some of that data. Again, Max Fort, Multi-Fort, Dero, 141TX. We've had good luck with Fort Amino so far. I mentioned that one in just one slide. It's a pretty new rootstock, and Cardia as well. But we still have a lot of work to do to look at how that affects the nutritional quality and the organoleptic or the flavor of the fruit as well too. So like I said, we're submitting a proposal tomorrow with a big group, and then also we're submitting another one here in about a month that looks in even more detail at that nutritional quality stuff. I think, the whole thing with grafting is best put by one of my friends here in Kansas who says, why would I build another high tunnel when I can just use grafted plants? And there's something to be said about that. There's certainly, some money invested in these plants when you do it. But if you're able to increase your yield by 20, 30, 40, 50%, it's a nice way to add some more dollars into your pocket. So the last thing I just wanted to wrap up with was just to let you know what we're doing. Literally today, actually tomorrow, we're submitting this proposal to NIFA. And again, this is a collaborative project with a whole bunch of universities around the country that I think are sort of leading the world of grafting. And I sort of outlined the research and extension objectives. I'm not going to read them all there, but we're doing some unique stuff this time. One is working with Carnegie Mellon University, who's got some really cool technology trying to develop robotics for propagation. But then probably more important to us as growers, the real thrust of that proposal is really looking at optimization. So, sort of recognizing that people are either using grafted plants or they're not, and how you manage those plants can really affect their performance. In the same way that we saw the benefit of using those grafted plants go up with production potential, you can apply that same idea to a whole bunch of different things, So I'm really excited to get that work going, and fingers are crossed that it gets funded. And then, hopefully for y'all on the extension side, we'll have some good deliverables. We've been in communication with the folks that write the commercial production guides, and we're hoping to just put our rootstock info straight into those so that you can get a little bit more grafting information through those. And then again, I mentioned that apprenticeship before. If you know anybody that's growing plants in a greenhouse or interested in learning more about grafted plants, please tell them to reach out to me, because we're really, really interested in making that more of a formal program. So with that, I apologize. It was a ton of information, and I really appreciate you all paying attention through it. And if there's time, I'd be happy to answer any questions.
Okay, thank you very much, Cary. Do we have any questions? Yes. So, Cary, the question was, and make sure I'm getting this right, so what were your assumptions for creating the production curve? Okay.
Right, for those, where we took all that sort of meta-analysis where we put it all together?
Yes, yes.
Yes. Well, I mean, I guess I'd be happy to chat with you more about that sort of afterwards. But, the basic assumptions that we had to follow was the scion type, whether or not it was grown in a high tunnel or a field. Those are things that we kept separated. We did not separate organic and conventional data because anecdotally, at least, it doesn't seem like it makes that much of a difference typically in the world of grafting. And then, basically being able to make the assumption that we can put that data together. And that's the part that we're still sort of working with statisticians to make sure we're doing that appropriately.
Okay, very good. Any other questions? Yes.
Cary, what was the condition for healing chamber?
The question is the condition of the healing chamber?
Sure, yeah, I didn't even get into the details of how we produce the grafted plants. So there's a lot of information out there, so I definitely suggest using old Dr. Google to help with that. But what we like to do, it depends if you use leaf removal or not in terms of the length. If you don't use leaf removal, it's about a 7-day process. If you use leaf removal, it's about a 10-day process. And all of it is a little weather dependent when you're in the greenhouse. So I'll start with those two disclaimers. But generally what we're looking to do is have as high a humidity as we can, 95% plus for about the first 72 hours. And then you want to reduce the humidity a little bit, but not a lot, because if you keep it real high, then you're going to have problems with adventitious roots. But if you get too low, then the plants are going to wilt down and you're going to have issues. Typically, those plants take 7 to 8 days to actually do the physiological work of healing. And so that time between day 3 and day 7 is really critical for sort of like walking that line in terms of humidity and light. And typically what we like to do is reduce the humidity just a little bit, 85%, 80%, not below 80. And then really keep a close eye out for the sun. And that's something I've been learning just in the last few years. If it's been, there's nothing better for growing grafted plants than snowy weather. Because when it's cloudy and snowy, there's no stress on that healing chamber. Those plants, they just grow and they heal, and you never see them wilt. And you just pull them right out, and it's like it's magic. But when there's sun out, and especially if you go from a cloudy day to a really bright sunny day, then all of a sudden the plants in that chamber will freak out. And even if you have just little slivers of light that come through the sides or the corners, you'll see those plants that get hit by that light will start to wilt. So you have to really watch the light conditions really closely and just be able to, throw shade cloth on them. Again, we use that foam board, can be really helpful. But that 3 to 8 day window, you sort of want to back down a little bit on the humidity. And then again, around 7 or 8 days, that's sort of when you start to unwrap them and get back to sort of more ambient conditions.
Okay. Was there another one? Yes. Yeah, so there's a question in the audience about someone interested in maybe purchasing some grafted plants from you, and how would that work?
Sure. So we do that all on an order by order basis, in that we don't have, I don't have plants sitting in the greenhouse ready to be sold. It's a custom grafting services thing. So the first thing you do is you just reach out to me through email. There's a person that works at my vegetable technician basically runs that program. And so I'll probably pass your info along to her. But essentially what you would want to do is get us the seeds at least 8 weeks before you want to pick them up. And then the idea is we don't guarantee anything, but we find that 8 weeks is about the right amount of time from when we seed them until they're ready to be picked up. And so essentially Cassidy, we make a schedule starting around Thanksgiving, and we actually base our whole schedule on 2,000 plants a week. And so it's sort of first come, first serve in terms of when you would get yourself on that schedule and able to get the plants exactly when you want. But other than that, it's just getting us the seed. And this year we charge $1.50 per plant in order to do the grafting. And that basically, again, you have to buy your own seed, and you will need to buy more seed than you want in plants, because they aren't all going to germinate and not all of the grafted plants are going to be successful. But yeah, that's pretty much how it goes. And we start lining that stuff up typically around Thanksgiving.
Okay, and then they would drive out there to pick up the plants?
Yep, yeah.
Okay, all right, very good. Sean?
There's a team of us working to have grafted tomato plants right up the hill here
Very good. Thank you for mentioning that. Yeah. Yeah. Any other questions? Yes.
A little more about what your expectations are for your propagation entrepreneurs, the ones that you're trying to get on the grant.
Yeah, the apprenticeship. So, one thing I will say is that, so we're submitting that proposal tomorrow. It may or may not get funded. And even if it does, it's going to be at least another year before we really get that thing going formally. But I'd be happy to start on a more informal basis before that. But the idea is basically to find greenhouse growers in the area that, have a green thumb, quite frankly, and the facilities to be able to grow grafted plants, and then link them up with us. We can train farmers to do it, but most of them don't want to. And, we just find that these greenhouse operators, we've actually published economic studies that show they make more money by growing and selling grafted plants than they do non-grafted ones, just because of that higher markup. So the idea is they would basically take online courses with me in terms of how to do it. And then we're planning to set up a virtual apprenticeship. So have a little gimbal camera that I bring into the healing chamber room every morning when I'm doing healing chamber checks in the greenhouse, and show people what how we do it, essentially.
Okay, very good. Well, that is the end of our time together, so thank you for joining us, and I'm sure you're going to have some follow-up questions from the audience, so thank you very much.
Sounds great. Thank you all.
Let's give you my